Get it? Like I was so kindly introduced, my name is Kyra Johnson, and I'm currently a doctoral student at Rutgers University in New Brunswick, New Jersey. Today I'm going to present part of my dissertation project or preliminary results from my dissertation project that I've titled A Revised Approach to the Use of Cortical Surface Texture of Bone for the Identification and Interpretation of Heat Alteration. Briefly, I just wanted to start by going over the importance of fire in human evolution. So fire has played a major impact on morphological and behavioral evolution, including being hypothesized as being one of the major drivers behind our increased encephalization, the decrease in molar size, as well as humans' ability to expand and alter their habitats. Unfortunately, despite this being an incredibly important tool, fire residues, specifically the diagnostic ones like hards, are fragile and are often destroyed by post-depositional processes, making it hard to detect in the archaeological record later. There are a multitude of ways or proxies that we can use to study fire, including other sediments outside of a hearth structure, plant remains, lithics, and bone. Today, specifically, I want to focus on bone and the way it can be interpreted at a multi-level resolution. So we can look at anything from the way the color changes to the surface texture to the way that the crystal structures react in relation to heating conditions. Specifically, I am focused today on that surface textural analysis. This was first pioneered by Nicholson in 1993. Her experiments were done with sheep, pigeon, and fishbone samples that she burned at one uniform time, and then used a binocular microscope and scanning electron microscope to document how color and morphological features were changing on the surface of the bone post-burning. With these results, she was able to create 5 categories that could then be correlated to specific temperature ranges, similar to the Steiner et al. 1995 color stages that are traditionally used as part of fire interpretation of archaeological materials. These categories, though, are made or constructed with various different variables within them, and I am interested specifically on how all of those different variables that make up these categories vary more specifically with different burning conditions. So the variables that I was looking at today for surface characterization fall into 3 categories. The first one is these whole surface variables, so that would include the general texture, whether char is present, what that char looks like, whether there are layers, the color, particles on the surface, etc. But we can also look at more specific features on the surface as well, specifically cracks, so how much of the surface is being covered by cracking, what is the depth of those cracks, how which direction are they going, are they following the collagen fibrils within the bone itself, or what are the general shapes. As well, we can look at these vascular canals, which appear as pores on the surface of the bone as that initial very top level of cortical bone is destroyed. That once again can be characterized by talking about how much of the surface is covered by vascular canals, what are their shapes, what are their size, and do they have these little tails present that you can sort of see in this picture on the slide. So to do this, I have an experimental data set of 252 samples that I burned under variable conditions. I had 2 species that I was testing, so the American bison or bison, and white-tailed deer, Ordicolis vosgianus. And I had a set of each of these species that I burned while they were still fresh and wet, and a set that was dry. I dried them in a drying oven at 80 degrees Celsius for a month or until their weight plateaued to try and rid the matrix of as much water as possible. I also burned the bones at a range of unburnt to 200 to 800 degrees Celsius at 100 degrees Celsius increments. And then I also had 3 different times that I burned them at, so 30 minutes, 2 hours, and 12 hours. After all the burning was complete, I was then able to take those bones and take 3 pictures at 10 times magnification along the long axis of the piece. So the first thing I really looked at was, do we have species level differences? In the original Nicholson 1993 article, she was able to see species level differences or species level patterns in those surface texture features. Because both of the species in my study were mammals, large mammals and ungulates, we do not see a ton of variation between these species. It seems to be more of a order level difference rather than a species level difference. So here you can see the comparison between bison and white-tailed deer at 300 degrees Celsius and 400 degrees Celsius at all three of my times, and they're not very varying between the species very much. In my patterns of looking at the individual variables, I also see very little variation between, how much char is present and how much cracking is present. And then after I looked at the species level, I was interested in understanding whether the condition of that bone at the time of burning impacted these variables. What I was seeing, once again, is not a ton of variation. Once again, I'm showing on this slide the presence of char and the presence of cracking. The dry and fresh samples do not seem to have very, very similar patterns between the two conditions. It seems like the majority of variation in this case is coming from temperature. On this slide, you will see an example picture from my bison samples that were burned for 2 hours at each of my temperature intervals. And so we can see with these temperature intervals, we're getting a lot of variation in not only color, but also the overall texture, the presence of char, amount of cracking, and those little vascular canals that we're seeing specifically at 500 and 600. So once I figured out that the majority of variation was coming from temperature, similar to what Nicholson was seeing in her original study, I was interested to see how much time played a role in the variation of these features. Here on this slide, you can see that I am displaying samples from 300, 400, 500, and 600 degrees Celsius at all three of my time intervals. And this temperature range seems to be where we're getting the most variation in our morphological surface features. Specifically, 400 degrees Celsius has a lot of change in between 30 minutes and 12 hours of burning time. And we can also see these patterns carry into some density maps for some of these features. So once again, I'm showing char and the presence of cracking. Char in particular seems to be one of the better variables that I was looking at for determining between different temperatures, different times, etc. So here you can see that really at 12 hours, it pushes the presence of char much lower temperature wise than we see in the 2 hours and 30 minutes samples. And then also in presence of cracking, we see that cracking is typically, or its absence of cracking, I should say, is pushed lower, whereas the 30 minutes is pushed a little higher, or it persists a little longer. So I propose that the reason we're seeing the majority of variation happening at that in those middle temperatures from 300 to 600 degrees Celsius is because of the dramatic change in composition that is happening during these temperatures. As you can see in this figure on the slide that I'm showing, this temperature range is an incredibly important period for changes in the organic composition of the bone. That organic material is being combusted and removed. And then as that organic material is being removed, we have a change in the crystalline structure because the crystals are changing or growing and reorganizing themselves, therefore impacting the overall structure of the bone and sort of degrading how it's able to hold itself together. In the future, I really hope to refine these variables for easier and more accurate classification. Some of the variables that I used for this study had a lot of different subcategories that I was using to classify, and I think that overcomplicated some of the readings. In addition, I hope to complete some discriminant correspondence analysis to help and identify whether there are variables varying together rather than just looking at the variation individually. And then eventually I have samples from 3 sites in the Iberian Peninsula that I hope to apply these results to. And I also hope to combine these results with results from colorimetry and FTIR data from the same experimental samples that I'm doing for my dissertation. And thank you.